Okay, hello, welcome to El Zoominario. Today we're very happy to have Federico Rey. Federico is from Villa Maria, Cordoba, Argentina. He got his undergraduate degree in biochemistry at the Universidad Nacional de Cordoba in Argentina. He got his PhD in microbiology at the University of Iowa, and he did a postdoc at Washington University. Currently, he's an associate professor at the University of Wisconsin-Madison. Thank you so much, Federico. We're very excited to have you here.
Thank you, Claudia, for the introduction, and great to be here. Thank you for the invitation. And yeah, so this is I'm here in Madison, Wisconsin, and beautiful picture of a picture of a beautiful Madison and the lakes that we have. And as Claudia mentioned, I'm originally from Argentina, so I'm going to repeat what Claudia said, but with pictures. So I'm from so small town called Villa Maria that is in the state of the provincia de Cordoba, and and actually this area in Argentina produces as much milk almost as Wisconsin does. So it's an area with a very strong dairy industry, dairy production of milk products, and actually I did my I had no plans to come to Wisconsin, but I did my high school was I got a technical degree in dairy industry. And so this is me here making cheese. During this year, in this technical school, I learned a lot of chemistry and biochemistry, and really this was a motivation, motivated me to wanting to learn more, biochemistry basically. And so, but we also had a good, good time. And so this is still me in high school, and this is, this is my car I drove, in my teenager years. And here's with a band that we play a couple of times in high school. And so this this exposure to making cheese and so on motivated me to start study biochemistry, and got a bachelor in biochemistry from the Universidad Nacional de Cordoba. And so after finishing that, I came to the U.S. and came I actually work as a scientist for a couple of years at Henry Ford Hospital. This just doing research and before I applied to grad school. And so when I applied to grad school, I actually applied to very different to maybe what people in general do here. So coming from Argentina, I was completely clueless of the process. I only applied to 3 schools, and the only school that took me was University of Iowa. And I have to say that I worked 2 years at Henry Ford Hospital with Patrick Pagano, and we published. So I published 3 papers as first author. And even with those 3 first author papers, the only place that took me was University of Iowa. And I started my PhD at University of Iowa, and I did my PhD with Kerry Harwood in bacterial physiology. Kerry moved to Seattle, so the University of Washington, and so I finished my PhD there. And so at that time, I completed my PhD in 2006. And during the last year, I spent a lot of time thinking about what I was going to do with my life. So my work with Patrick Pagano was on cardiovascular disease. During my PhD, I completely shift gears and fall in love with bacteriology and study bacterial physiology by this photosynthetic bacterium that could generate hydrogen gas from pollutants and phenolic compounds. And so really spent a lot of time in my last year of my PhD thinking what to do with my life and for postdoc. And I I came across these 2 papers published by Jeff Gordon. So this is the early days of the gut microbiome. And so this one with Frederick Bachelet as the first author, suggesting that the gut microbiome could influence obesity. And then this other paper, Justin Sonnenberg, showing how bacteria could eat mucus basically produced by the host. And really this really inspired me to read more about the gut microbiome, learn more about my microbiome. And I was very lucky to get a position in Jeff Gordon lab, University at Washington University, where I did my postdoc. And so, sort of, I started working with him, since 2007 until now. I studied the gut microbiome, and this is sort of what keeps me up at night, sort of thinking about the gut microbiome. And so I'm going to tell you a little bit about the sort of the work that we do in my lab. I'm going to give you some general intro about the gut microbiome and some of the work that we do in my lab. And so I'm sure sort of by now everybody has heard about the gut microbiome. It's sort of unlike sort of before before I started this, now we every day seems like there's a paper on the gut microbiome. And so we think about the gut, sort of we know that our gut our intestines are populated by trillions of microorganisms, and that each of us, and these organisms actually sort of span the 3 domains of life. So we have a lot of different bacteria that live in our gut. We have some archaea, few members of the archaea, and we even have sort of some eukaryotes. And so each of us harbor about 100 to 200 different species of bacteria, and the abundance of these organisms can shift quite rapidly in response to diet. But sort of like these organisms, we start to get colonized, as we come out the birth canal, and sort of by the time we're sort of 3-4 years old, so we have assembled a community in our guts that is quite stable, sort of as far as we don't make sort of very profound changes in diet. And so it's relatively stable as far as we can tell. You can see this very similar populations or communities sort of as a function of time for a similar individual. But then what we also know is that it's quite variable across different individuals. So each one of us has a unique collection of bacteria, archaea, and eukaryotes. And so again, sort of here, sort of one consequence of having these microbes is that they help us digest our food. Everything that that we consume, they get to interact with before our sort of Homo sapiens cells sort of do. And sort of coming from Argentina, when I think about food, this is sort of the first thing that comes to mind. And so every sort of simple, every component of this diet will be processed or in one or another way by gut bacteria. So for example, if we look at the salad, sort of this play here, we look at the salad. The salad is a rich source of plant polysaccharides. These plant polysaccharides are food for gut bacteria. We humans or mammals in general, we lack the enzymes to process this salad. And so we rely on our gut microbes to break it down and turn it to compounds or metabolites that our body can sense and sort of unprocess, like short-chain fatty acids, metabolites like acetate, propionate, butyrate. And so these, short-chain fatty acids sort of serve, as they contribute a little bit to calories, but more importantly, they have very key signaling functions in our body. I mean, we have specific receptors to sense and respond to these molecules, and those receptors are in all our, sort of many different parts of our body, including our brain. So we have some of these receptors. And so these microbes, by means of making these short-chain fatty acids, they modulate the influence of our biology. These short-chain fatty acids have immunomodulatory properties. They influence sort of motility of the intestine. They influence sort of how we deal with glucose, how much insulin we make. They regulate our blood pressure and expression of our cells. And so all these are just, from this salad that we're here, that we are providing substrates for the bacteria in our gut to make these, sort of these compounds. And the amount, sort of the proportions that each of us will, or each of our microbiomes will make from this will vary largely as a function, as in, of each person. So if if everybody that was in the call will to get this the same salad, the abundances of acetate, propionate, or butyrate that will be detected will vary. And so likewise, sort of this glass of wine is a rich source of polyphenols, or these antioxidants that are supposed to be in sort of red wine. But it turns out that a lot of these antioxidants are not absorbed as they are present, in the wine. In fact, they are some of them are heavily metabolized into compounds that are way more potent than the compounds that are in wine, and way more bioavailable. And these compounds can all influence things like cardiovascular disease, protect from cardiovascular disease. What's interesting is that everybody, again, if everybody was to have this glass of wine, everybody would generate different abundances of this. Will generate different levels of these metabolites. And then sort of like if we focus on the meat, there are compounds that are like carnitine or choline or histidine that are present at relatively high levels in beef, sort of, and they are important for us. But it turns out that there are bacteria in our gut that can also use this compound and generate metabolites or compounds that may not be as good for us, and may contribute to the detrimental effects of beef to health. And so I'm going to talk a little bit more about that sort of later in my talk. So sort of the idea is sort of that there's these large interpersonal differences in the composition of microbiota. And so my lab is interested in understanding how our microbial differences modulate the nutrients that we get from the food that we consume, and how this affects our health. So the central hypothesis that drives research in my lab is that the nutritional value of food is influenced by the collection of microbes that are present in our gut. So we think that if we can understand that, we can sort of personalize nutrition based on the collection of microbes that we have in our gut, and based on what the microbes can do. But we still don't fully understand what they can do. So and who's doing what. So this is sort of part of what my lab is trying to do. So in trying to understand sort of these interactions between food and microbes in our guts, we're faced with the challenge of the high complexity of the system. I told you that each of us has between 150 to 200 different species of bacteria, and sort of there are a lot of microbial genes in there to study and understand. So these communities have they're very complex. And so one approach that my lab uses is to sort of colonize germ-free mice that are representative, colonizing with microbial communities or bacterial communities, mostly, that are representative of native communities in terms of phylogeny and in terms of function. And so by using this approach, we eliminate sort of like the uncertainty surrounding the sort of who's there. So we can we take these mice that are germ-free, and we introduce specific bacteria that we're interested in study. And and in some cases we can manipulate genetically the species bacteria that we're introducing. So we can really manipulate the system to test hypotheses. So here's sort of like the two tools, main tools that we use in my lab. One is the what I'm showing you here, to the left is and so maybe I'll to the left here is an anaerobic chamber, and inside this chamber we grow a lot of the bacteria that we use to colonize mice. So a lot of the microbes or the bacteria that colonize our gut are anaerobes. So they're actually sensitive to oxygen. So if they're exposed to oxygen, they die. So they grow under anaerobic conditions, and we create anaerobic environments in this chamber to grow a lot of these bacteria. And the other tool that we use sort of frequently is sort of these germ-free mice that I mentioned, that we raise them in these plastic bubbles, and everything that we put in these bubbles has been previously sterilize, in some way, like either using, an autoclave, sort of like, or chemically sterilize. And by using these tools, we are focusing on sort of trying to understand how, microbes modulate sort of progression of metabolic diseases, cardiovascular diseases, and in the last few years, we have developed an interest, and sort of we are exploring how the gut microbiome impacts progression of Alzheimer's disease. So just sort of like, in this time that I have left, I'd like to give you an example of, sort of how we are tackling these questions or these issues, sort of like, and so what I'm listing here some of the papers that we have published over the years on, studying a specific interaction between gut bacteria and a nutrient that, is of interest to us, that is choline. Choline is an essential nutrient. We need to consume the, choline, but we also sort of have bacteria in our gut that, like choline, and they will use choline for their metabolism. And as as an end product of their metabolism, they will generate compounds that are not necessarily good for us. And so this is sort of what, we're studying. So, here's sort of choline. And, as I said, choline is important sort of methyl donor for a variety of biochemical reactions. And so, we need to consume choline from the diet. So we have some biosynthetic capabilities, but it's sort of like they're not enough to keep up with the needs. And we use choline to make methionine and methionine. And so, when sort of we need, we use that to sort of make methyl groups that we use for DNA, RNA, sort of for lipids and proteins. And so, but it turned out that there are bacteria in our gut that, as I mentioned, sort of like they like to metabolize choline, and they will take, choline from our diet, and processes will convert it into a metabolite called trimethylamine. And so trimethylamine is a gas that can enter our circulation, and then in the liver is converted to trimethylamine oxide. TMAO, so we're going to refer to trimethylamine oxide, is going to refer to TMAO, has been linked with a number of diseases, including cardiovascular disease, obesity, heart failure, diabetes, liver disease, colon cancer, and sort of the list keeps on growing. So it seems like this is not a good metabolite. And so, and something that we were able to demonstrate by using germ-free mice is that the gut microbiome really influences how much TMAO we accumulate or mice in this case can accumulate in blood. So here what we did is we took fecal samples from different individuals and put it into germ-free mice. So we engrafted the microbiomes from humans into mice, and then after colonization, we look at how much TMAO these mice have in their blood. And we can see that, depending on the microbiome that we use to colonize the mice, you can see no TMAO production on a ton of TMAO production. So, if you are here in sort of this, if your community has, no capacity to make TMAO, you might be good, you can eat all the choline you want. But if you are here, maybe like, sort of like this is sort of getting to personalized nutrition, you might restrict your choline consumption to levels that, just barely what you need. So, sort of we apply this neurobiotic system to try to understand the impact of bacterial choline metabolism on host biology. And the way we did this is sort of like trying, we first, sort of look into what microbes or what bacteria make TMA. Convert choline into TMA. So we, in our lab, we have a large collection of human gut isolates that we can grow in 96 well plates under anaerobic conditions, and with labeled choline, and ask which microbes can convert choline into TMA. We do that using mass spectrometry, and sort of we can, and by doing this, we found that from the bugs that we screen, we identify a number of species that were able to convert choline into TMA. So something that we learned is that it was a relatively small fraction of bacteria that had the capability to convert choline into TMA. So we got lucky that one of those bacteria that sort of had this capacity to make choline was E. coli. And so E. coli is actually sort of is genetically tractable, so we can make mutants. And so it turns out that what I'm showing you here is these are the genes necessary for making TMA from choline. So this is called the choline utilization gene cluster. And, we can, because we are working with E. coli here, we can create mutants in which sort of we delete the gene that encodes from the enzyme sort of required for converting choline into TMA. And so we did that. And so then when you make that mutant, and compare it to the wild type strain, so you can see that the wild type strain will make TMA from choline, but the mutant is dead. And something that I want to emphasize is this mutant grows under, fine with glucose or other, sort of substrates, but it's impaired for metabolism of choline. So it does not make TMA from choline. So this is sort of very useful to us because now we can go back to the mice and we can, sort of introduce these strains and ask, how does this pathway contribute to disease progression? So what we did is, we went back to our germ-free mice and we colonized them with a community of, bacteria that, sort of we're going to call this community cord, a core community consisting of bacteria that cannot metabolize choline and cannot make TMA. And then we divided those mice into two groups, one group in which we colonized with a wild type strain, and another group in which we colonized with a mutant. So now we have, two groups of mice that are genetically identical, they're eating the same food, and they have the same community, except for one bacterium that lacks one gene. And the gene is sort of the gene necessary for convert choline into TMA and sort of production of TMAO. So then using this, we can ask what is the contribution of TMAO or, the TMA producing pathway to disease. And so, here sort of we're asking that question. So for example, here we're looking at TMAO, sort of the metabolite itself. So we see that mice colonized with a wild type strain accumulate TMAO in plasma, but the ones colonized with the mutant do not. We look at sort of number, I told you that TMAO was associated with metabolic disease, with obesity and cardiovascular disease. So we look at the number of biomarkers of metabolic and cardiovascular disease. For example, here we look at fat pads, sort of the fat that the mice sort of have accumulated. And we see that mice colonized with a strain that makes TMAO are fatter. They have higher levels of fat, not only sort of accumulate inguinal fat, but they also accumulate more fat in the liver, which is sort of a sign of sort of metabolic disease. And they also have higher levels of glucose. And so, this is consistent with the fact that TMAO has been linked with diabetes as well. The other thing sort of that we did, so we found out that TMAO sort of crosses placenta, and sort of like, the developing babies are exposed to TMAO. So we using the paradigm that I just described, we asked sort of whether TMAO, sort of being exposed to TMAO sort of during pregnancy affected sort of behavior of, of mice. And so, here we colonize parents with the two communities, that I described. We wait until, they have pups, and then sort of, we look at the behavior of those pups. And sort of in this, behavioral assay, we look at the amount of marbles that the mice bear. So this is marble bearing assay, which test for anxiety is one of many assays that people use to test this. And so what we found was that mice that were colonized with the wild type species, then that they make the TMAO, they pretty much buried all the marbles that we put it in the cage. You would expect the mice to bury some, but here we're looking at the mice were going crazy and burying marbles. So we noticed that there was something sort of different with their behaviors, which prompted us to look at the brains of these mice. And sort of we found that there were differences in patterns of methylation in the brain of these animals. Not so much in the liver, we saw some. And so this sort of pattern of methylation, this is a way, methylation are epigenetic modifications that can happen in the DNA that sort of can regulate gene expression. So we don't know exactly what was going on, but this has, prompted us to, sort of like wanted to learn more about whether TMAO was affecting, neurodegenerative diseases or, metabolism in the brain. And so in order to sort of get at that, we started to collaborate with Barbara Bendlin. She's in the Wisconsin Alzheimer Institute here in sort of here in Wisconsin, in the university. And so we actually look at levels of TMAO in cerebrospinal fluid from healthy subjects with mild cognitive impairment or with Alzheimer's disease. And so what we see is that patients with Alzheimer's disease have significantly higher levels of TMAO in the cerebrospinal fluid. And we also see that the levels of TMAO in the cerebrospinal fluid correlate with biomarkers of Alzheimer's disease. So we're really interested into this, and sort of we have, this is sort of something that we're actively working on. Sort of we and others now have shown that feeding mice TMAO or colonizing mice with communities that make TMAO, like I described, exacerbates, markers of disease or signs of disease at least in mice. And I think sort of this, I'm going to sum up sort of what I told you so far is, so I told you that, we're using these non-biotic mouse models to, study interactions between bacterial metabolism and diet and disease. We have learned that there's a wide variation in sort of bacterial choline metabolism, interpersonal differences. And we've found that sort of this, this is something I did not talk very much, but we have found that this the pathway involving choline metabolism is discontinuously distributed among gut bacteria. For example, I told you that there was an E. coli strain that we work with, and that had this pathway necessary for making choline, but it's one out of sort of 500 different species, 500 different strains of E. coli. So most E. coli strains will not make TMA from choline. So there's quite a bit of variation even within the same species. I sort of told you that interactions between choline and gut microbiome influence sort of signs of cardiometabolic disease. And I show you fat, glucose. We see sort of changes in behavior, and we see some associations with with Alzheimer's disease in humans, which we recapitulate some of that in our mouse studies. And so, sort of, I think sort of a conclusion of this sort of is that sort of this TMAO is production pathways, a differential factor that modulate host responses to dietary choline, and a target where people are developing interventions to try to sort of prevent TMA or TMAO production when sort of we consume choline. I think that's sort of what I have for my science part. And then so should I
yeah, you can continue, Fede.
Okay, yeah. So I sorry, okay. So I'm originally from sort of say, sort of Claudia say, and I sort of mentioned, and I did. Oops, sorry. I'm originally from Argentina, and so I really I sort of don't know how much people know about Argentina. And sort of like what I think sort of many people get taken by surprise is when I remind them that now in Argentina is the end of summer, and everybody goes like, what? Wait. Yes, sort of we is in the South Pole and or the South Hemisphere, and you know, so we have inverted seasons. So now is the sort of getting to the end of summer there. And then Argentina is actually a sort of beautiful country that has a lot of, you know, beautiful areas to visit. Some, you know, sort of here I'm showing you. So this is sort of like some pictures of, you know, so like the bottom pictures are from the south. So a beautiful glacier, you know, sort of called Perito Moreno. And this is sort of an area called Villa Langostura, sort of beautiful, you know, so this is, you know, the Patagonia region, sort of. This other picture here is, you know, close from where I grew up. This is Cordoba, the provincia de Cordoba, where they are like these sierras, you know, sort of, you know, small mountains and a lot of, you know, nice streams of water, you know, very clear water, you know, beautiful area to relax. And then, you know, sort of like in the northeast, we have these sort of Iguazu Falls that, you know, are in the limit with Brazil. It's another beautiful area to visit. So, you know, I sort of, one sort of comment is, if you never thought of Argentina as a travel destination, it's sort of like, I highly encourage you to do so, because I mean, it's far though, but it's beautiful. It's worth the trip. We have good food too, but sort of the piece of culture that I thought I would share with people today is sort of a rich music, a great music that, you know, this country has. And I sort of wanted to provide folks with, you know, sort of names of, you know, sort of musicians that I really admire and listen to, and, you know, links to their Spotify sort of pages, sort of to listen to, or just check them out. So I'd like to start with Astor Piazzolla. So Astor Piazzolla is sort of he was he played tango music, but what's sort of really special about him is sort of that he was one of the sort of creators of this nuevo tango, sort of new tango music. He played this instrument, sort of that is in English, in Spanish, called bandoneon. I don't know sort of how you say that in English, but he was really amazing. But then sort of what he did really sort of creative, sort of he incorporated elements of jazz into his music, and that made it really unique. Sort of his, I think what I would rank him as probably our most prominent musician, full stop. He was very successful. He, you know, his music is played all over the world. Yo-Yo Ma, sort of like big cello player, sort of like plays his music. It's sort of like this one musician that if you haven't checked it out, I really encourage you to do so. It's really unique music. And then sort of we have a really rich, Argentina has a really rich rock history. I think sort of that, you know, sort of starting from the, you know, sort of late 60s, you know, sort of with, you know, a few artists that I think they are amazing. And if, you know, I think if they were born in the US or UK, they'd be the Rolling Stones. This is my very biased view of it. But I really encourage sort of to check them out. Sort of Charly Garcia, sort of it's more of it's not hard rock, it's sort of type of more rock and pop, and so I'm more like, you know, sort of something like Queen or Elton John, but it's really creative music, really amazing. Luis Alberto Spinetta, sort of he also was unique in that, you know, he sort of played had various bands in his career, but he was also sort of fused rock with elements of folk music and tango music from Argentina. His music is really amazing. And, you know, these two folks, you know, Charly Garcia and Spinetta, you know, sort of they were really famous in the 70s and 80s. So I don't think that they reach, you know, out much of, you know, outside of Argentina. So I don't know, sort of in other, you know, countries, you know, in Latin America are famous. I mean, I know that, you know, some people know them, but I don't know sort of the level of fame that they got outside Argentina. I don't think very much. But sort of I know that Soda Stereo, so it's a band that, you know, much younger. I mean, like sort of they are now, they have sort of, they're anymore, sort of the lead singer died a few years ago, but that they were really prominent in the 90s, and they really existed. I sort of always think that the reason they became sort of the rock scene sort of became so big in Argentina was because of these two guys that are really amazing and paved the way for all these other bands, including Soda Stereo, that really made it in the international scene. And sort of Soda Stereo sort of played in Mexico thousands of times. I think they were as popular in Mexico as they were in Argentina or anywhere in South America, Central America. So I think that's sort of all I have, and I'd be happy to take any questions. You know, sort of I'm not as foodie. I mean, sort of like I'm very limited on my food consumption, sort of to beef and barbecue. So I thought I would share some.
Thank you, Fede. I'll stop the recording and then we can move on to questions.